Hello, this second video will be on organelles. You're going to create a gigantic animal cell. So if you have a big piece of paper, write it on a big piece of paper. If not, put your paper in landscape view, and we're going to draw out a big animal cell that we will add more detail to in class on Friday. So at the top, I'm going to my animal cell. And then I'm going to draw a gigantic cell membrane. That takes up most of the page. So remember, in a cell membrane, a cell membrane is made up of phospholipids, right, with embedded proteins. So whenever I'm drawing this line for the cell membrane, please remember, and I'm taking up the entire page, please remember that this entire line represents a phospholipid bilayer. So this entire line represents the cell membrane. And we remember that all cell membranes are biomembranes made up of phospholipids with different embedded proteins. And also, of course, here's my embedded protein. And also, of course, other things like cholesterol to maintain the fluidity of the membrane. So whenever I use this blue, please know that it is a biomembrane. And remember, an animal cell is a type of eukaryotic cell, which means there are internal membrane-bound organelles. Okay, so we're going to draw a very large nuclear membrane, which, as we remember from our previous video, is also a biomembrane. Then we'll draw a very large endoplasmic reticulum, which also, as we remember from our previous video, is a biomembrane. We're going to make this the rough endoplasmic reticulum, or rough ER, which means that it has ribosomes on it. Okay. Over here, we'll add in our smooth endoplasmic reticulum. And I'm just going to call that our smooth ER, which does not have ribosomes on it. It's bigger, it's probably more in line of the rough ER size, but this is not to scale. Okay. And then farther away from the nuclear membrane, and remember the smooth ER is also a biomembrane, so it's made up of a phospholipid bilayer with embedded proteins. Then farther away from this rough ER, we have our Golgi, which is also made up of phospholipid bilayer with embedded proteins. So the Golgi is also made up of a biomembrane. So we have the nuclear membrane, the rough endoplasmic reticulum, and the Golgi. We might have some small vacuoles, again, which are biomembranes. We might have some lysosomes, again, which are surrounded by biomembranes. So all of these organelles in eukaryotic cells have membrane-bound organelles. They also have ribosomes, which aren't membrane-bound organelles, but most of them are membrane-bound organelles. And finally, I'm going to add in this corner over here the mitochondria. It's not very pretty mitochondria, but it's the mitochondria. The mitochondria is interesting because it has not one, but two membranes. Because remember, this blue color is showing you biomembranes. So the mitochondria has an outer biomembrane and an inner biomembrane. Okay, so these are some of the membrane-bound structures in a eukaryotic cell. Remember, we also have the cytoplasm, which is the fluid-filled section of the cell. We have the cell membrane, as we said before, and we can have the cytoskeleton, which I'm doing in a different color because it is not a biomembrane. The cytoskeleton is a series of microtubules and microfilaments that help provide structure inside the cell and help move things inside the cell. So inside the nuclear membrane, right, this is the nucleus right here, we also have the nucleolus, and the nucleolus's job is to make ribosomes. So we'll do that right here. And it's not membrane bound, it's just a region in the nucleus. So this is the nucleolus. The nucleolus makes ribosomal subunits, which means it makes parts of the ribosome. Okay, those parts of the ribosome then leave through holes in the nuclear membrane. These holes in the nuclear membrane are known as nuclear pores. So the nucleolus makes ribosomal subunits. These ribosomal subunits move through the nuclear pores, assemble themselves, and become ribosomes. Also in the nucleus, we're going to find the chromatin, or the DNA. Right? So here's our DNA. And when it's this kind of stringy format, it's called chromatin. Okay, just type of DNA. So in the nucleus, you're going to find the DNA, and in the nucleus, we'll go through the process of deciding what part of the DNA we want to read. We're going to read a section of the DNA, and we're going to create mRNA out of that section of the DNA. And that process is called transcription. And then that mRNA is going to be carried through the nuclear pores, and it's going to attach to one of the ribosomes. So here we have our ribosome. Here we have our mRNA. And then our tRNA is going to come in, and we're going to end up making a protein. And that process right there is called, where the protein is made, is called translation. In class, next class, we'll pick up in the cell and we'll talk about how this protein can become a membrane protein, how this protein can end up in the lysosome, or how this protein can be exported from the cell. And it's all due to the way that the ER, the Golgi, and these little vesicles and the cell membrane work together. And the lysosome will come into play as well. Okay, so make sure you have this diagram ready for next class, and we'll add to it. Thank you so much.